Populations rarely, if ever, live in isolation from populations of other species. In most cases, numerous species will interact and live kind of interconnected with one another to share a habitat. Now, the interactions between these populations, they play major roles in regulating population growth, size, composition, and abundance. Now, all populations occupying the same habitat form what's called a community. Communities are naturally occurring assemblages of species populations that inhabit a specific area at the same time. Right? So there are these groups of populations of different species that occur together across space and time. Now, ecology is often studied at the community level in an effort to understand how species interact with one another and also how they compete for similar or shared resources. And not only is it studied at that level for understanding species interactions, which is definitely a major factor for such, but we also study communities because communities in and of themselves are pretty interesting. Different ecological communities can be pretty different from one another in terms of the types and numbers of species that they contain. And one way to describe this difference is to say that communities have different structures. Community structure is essentially the composition of a community, including the number of species in that community and their relative numbers. Now, the structure of a community is influenced or can be influenced by a variety of factors, including such things as disturbance, species interactions, chance events, and abiotic factors. Now, as far as describing or understanding community structure, there's two hypotheses or two types of hypotheses that exist for doing such, as well as for kind of describing how communities are modeled. These two hypotheses are the individualistic hypothesis and the organismic hypothesis. The individualistic hypothesis is also known as the continuum view. This was created and birthed by a man named Henry A. Gleason. Now, Gleason believed that a community was merely this fortuitous association of organisms whose adaptations enabled them to live together under the particular limitations of that environment. So these associations of organisms were more of just coincidence. It was by chance that they were able to associate with one another because they had those adaptations that enabled them to live together. It was just coincidence. He also believed that each species was distributed independently of others that co-occurred in a particular association, which is an organization that's often referred to as an open community. Now, open communities have no natural boundaries. There's no barriers to the movement of organisms, so organisms can actively emigrate or immigrate into or out of these communities. And the limits of open communities kind of in this same breath are arbitrary in respect to geographical and the ecological distributions of the species that comprise them, both floral and faunal species. Now, in an open community, because of these arbitrary limits and the lack of distinct boundaries, the distribution of species across the environmental gradient of that community occurs at random with respect to other species. So species are not clumped into kind of these active groups that are predicted or dependent on the presence of another species. That's Gleason's view. Again, this notion of open communities, that all communities operate in this manner. A lot of ecologists today favor Gleason's view, and they use Gleason's view of communities as being these, again, random assemblages of species as a means to support the concept that communities are not in equilibrium, that they are, in fact, these dynamic, influx regions or groupings of different species. Now, this is quite different from the view of the organismic or interactive hypothesis, which was kind of coined by Frederick E. Clement. Frederick E. Clement's view of communities was essentially that species in a community acted as interdependent superorganisms, hence the organismic model, as it's called. So the community itself is like this living, breathing organism. And this concept of the community being this organism was based on the belief that species belonging to a community were closely associated with one another. And they were also restricted to the confines of that particular community. So, for example, when you think about cattail water lily communities, they are restricted to bodies of water. You will see them continuously occurring along aquatic-based habitats. Now, these species assemblages were formed by closely linked species, right? And the species of these communities or these assemblages were locked into an association with one another by mandatory biotic interactions. And those interactions would cause the community to function as a unit. So essentially, these organisms formed a community because they were basically locked into the confines of an association because of the requirements they needed to uphold in order for their fitness and survival, but also the requirements of that habitat itself. So they're basically confined to that community and locked into that community to form an association. But that association allowed that community to function as a unit, hence where you get this idea that communities are assemblages that are based on the interactions of its members acting like a superorganism. So a little abstract way of thought, but it does hold substance. Clement also perceived communities as being these discrete units with sharp boundaries. In other words, species had distinct ecotones. An ecotone is a region, or ecotones are regions of rapid replacement of species along a gradient. Right? So you can think of like a logpole pine forest that you'd find in southeastern United States. that occurs in very dry areas. And it's distinctly different from a fir forest, which you would find growing in the cooler, moister habitats of maybe northwestern United States. So basically, stark differences between the species that create that individual community, as far as the floral and faunal species. Now, one of the important concepts of when we're talking about communities, a lot of it has to do with what species are present in these communities, where the vegetative and animal species that actually inhabit and make up or comprise that community. And this goes into an important concept or topic known as biodiversity. Biodiversity describes the variety and variability of all living organisms within a given ecological area. And it can be used to refer to the diversity as far as number of species. It can refer to the genetic diversity of those species or populations, or even habitat variety, so ecological variations. And it can be measured at and monitored at several different spatial scales, these scales being alpha diversity, beta diversity, and gamma diversity. Now, alpha diversity refers to the average species diversity in a habitat or specific area. So it's a local measure. It's simply looking or evaluating richness and evenness of individuals within a single specific habitat unit. On a little more of a broader scope of study is beta diversity. Beta diversity refers to the ratio between alpha diversity and regional diversity. So, kind of better put or more easy to understand, it's the diversity of species between two habitats or regions. And you can calculate it by taking first the number of species in, say, habitat 1 minus the number of species that are in common between the two habitats of interest. And then you also take the number of species in habitat 2 and you subtract from that the number of species that both of those habitats have in common. And then you can add those numbers together, and that gives you beta diversity. So you can use beta diversity again to evaluate the diversity of species between two regions of interest, but you can also use it basically as a rate of species turnover between habitats. On the broadest scale is gamma diversity. Gamma diversity, again, is the broadest of these spatial scales, usually occurring or considering landscape diversity, regional diversity, actually even beyond regional diversity, sometimes even global diversity. So it refers to, or it's monitoring the total diversity of a landscape, or again these large regions of habitats, and in general is a combination of both alpha and beta diversity. Really just the number of different species that are observed in all of the habitats within all of that, within that entire region or across that entire landscape. All right, so we can kind of put this to terms visually with some cute little fish. In this image, each of these, think of each color, each different color of fish representing a different species of fish, right? So your orange fish are one species. These icky green ones are another. The purple with black striped ones are another species. So we have three sites, three habitats, right? We have site A, site B, site C. We can calculate the alpha diversity for each one. Again, alpha diversity is that local measurement. So it's looking at the diversity of each one of these individually. So for site A, your alpha diversity is 3 because you have 3 different species. You have the black fish, which is one species, the orange fish, which is another species, and this icky green color, which is the third species. At site B, you also have an alpha diversity of 3 because, again, you have 3 different fish species. At site C, there's only 2, so that means you have an alpha diversity of 2. Now, beta diversity is comparing the diversity between 2 of these sites, right? So we can take the beta diversity between site A and site B. And to calculate that, we'll start with site A, right? So site A has, in total, alpha diversity of 3. So it has 3 different species, right? Now, between these 2 sites, how many species are common? Well, both sites have this orange species and the green species. So 2 species are shared between the sites, right? So from the alpha diversity of 3, you subtract 2. 2 are shared. You do the same thing for site B, right? For site B, we also have 3 species, right? The alpha diversity of 3. And there are 2 species that are shared between site B and site A. So to calculate beta diversity between these 2 habitats or individual communities, you would take 3 minus 2 plus 3 minus 2. That gives you a beta diversity of 2. So beta diversity between site A and site B is a value of 2. Now for site B and C, you approach it the same way, right? So for site B, we have 3 species of fish, right? And then looking between site B and site C, how many species are different? Well, let's see, site B has the green fish, and so does site C. But site C does not have the little red fish or the orange fish. And site B doesn't have the purple fish with the black stripes. So at site B, you have only one species that is shared, right? The only species that is found at both site B and site C is this little icky green guy. What about site C? Well, site C has 2 different species. Again, this purple guy and the green guy. Only the green guy is found at site B as well. So for the beta diversity, you take 3 minus 1, that's for site B, 2 plus 2 minus 1 for site C. It gives you a beta diversity value of 3. Now the gamma diversity is looking at the diversity of species for this entire ecosystem. Right? Now that's going to be a measure or value of 5 because there are 5 different species of fish that are occurring across all of these ecosystems or across this entire ecosystem. You have the orange fish, the black fish, green fish, red fish, and the purple fish with the black stripes. So that's how you calculate, again, these different spatial scales of diversity. Now, as I said before, with biodiversity, there's kind of 3 levels of biodiversity. You can look at genetic diversity, ecological variation, or the ecosystem diversity. You can also look at species diversity. Now, the number of different species occupying a biological community and the relative abundance of each species is known as species diversity. Species diversity is therefore a product and function of species richness and species evenness. Now, what the heck are those? Well, species richness describes the number of different species present in a community or within a given area. Right? So in general, the greater the number of species you have in a community, the greater the richness, the higher the species richness, right? So looking at these two communities, this community down here has a greater species richness than this community here because you have far more species types present than found here. Here you only have 3 species that are present, whereas, actually, sorry, you have 4. I didn't see this little guy here. You have 4 different species, whereas here you have 5 different species, right? So because you have more species here, you have greater species richness in this community. Now, species evenness. This describes the relative abundance of different species in an area, where if you have more species with more similar abundance levels, you have greater evenness throughout that community. Relative abundance refers to the population size for each species within a given area. So it's the proportion of each species. Right? So looking at this, the abundance of this white barked tree is significantly higher than the abundance that you'd find for that same species in this community. The relative abundance of this species in general in this community as well is far greater than the abundance levels of the other species, right? And we'll talk about this in a moment, why this is actually a poor indication of species diversity, why this community is representative of lower species diversity, whereas this is higher species diversity. Again, it's because it has to do with both the degree of species richness as well as relative abundance, right? Not relative abundance, with evenness. Evenness being a function of relative abundance, right? So if all of these species in this community, they all have relatively equal proportions to one another. They're relatively all equally abundant, meaning they all occur at relatively the same number of individuals per species in relation to the other species that inhabit that community. So as a result, because you have these species all have more similar relative abundances, you have a greater evenness in this community. So because this community has greater evenness as well as greater species richness than this community has a greater level of species diversity. Again, we'll go over that again in a little bit. But I do like to kind of delve into both of these components of species diversity a little bit deeper, just because there is a great deal of beneficial information to be gleaned that isn't necessarily provided by that superficial definition or view of each component. So we'll start with species richness. Species richness is the simplest measure of diversity. It is very simply just a count or number of different species in a given ecosystem, particular area or region. Now, often you will only count resident species, meaning you'll exclude accidental species from the count for richness. So you don't count species that are not native to that area. That may be like cultivar plant species that may be there by an accidental chance event. Now, when we talk about species richness, the richness of an area, as far as its species content, can be increased by speciation. Right? Speciation generates new species and therefore would add species to that community to increase or bulk species richness. Extinction will act as a counter to that. Extinction reduces species richness by removing species from a community. Now, there are some pros and cons to species richness as a measure. It's great because it caters to the interest in knowing the numbers of species in a community and how many species are there. It's also very easy to calculate and easy to understand as a metric. The problem with it is that it does not take into account abundance patterns among various species. So it ignores that evenness component of diversity. So again, this is why you don't evaluate or determine diversity by looking at species richness alone. You may have an area that has maybe 20 different species, but it's not going to be as diverse of an area if half of those species only have one or two individuals that are comprising them or representative of that species. Now, species counts themselves in general depend on the sample size or sample area. And this is demonstrated by species area curves. Now, in most ecological systems, the number of species found increases non-linearly with the area that is sampled, right? This relationship, per se, that exists between area and the number of species, it's very important. It has important implications for conservation biology and for different types of resource management strategies. Now, in general, what you find is that the number of species found or counted increases as you increase area. And there's two main hypotheses that have been proposed to explain that relationship, to explain that the species area relationship. The first argues that species numbers will increase in larger areas because they contain more types of habitats. So because they contain a greater habitat diversity, that allows for more places for species to exist, and therefore that increases species number and species diversity. The second hypothesis suggests that species number more is determined by this balance between rates of species colonization and extinction, right? So you can kind of think of these disturbance events. So it's a balance between new species being added and established in a community versus the rate at which they are going extinct. And we'll talk more about that in a little bit. But in general, this hypothesis developed from actually led to, I should say, the theory of island biogeography. The theory of island biogeography predicts that in the absence of all other variables, so not acknowledging the presence of invasive species or anthropogenic factors, forgetting all of those other external factors. A species, more species will inhabit or be found in larger islands than on smaller islands, because the so-called colonization extinction balance is reached when there are more species on those larger islands. And because those larger islands have greater area and greater size, there are more sites available at which species can inhabit. So there are more sites for them to colonize, but the sites are also large enough that you can have extinction occurring without extinction occurring too rapidly or not at all. So we'll come back again to that second kind of hypothesis in a little bit. But moving out of species richness into the concept of species evenness, which is that second component of species diversity. And a large proportion of species evenness is tied to this notion of relative abundance, right? It actually stems from this notion. In general, different populations in a community exist in relative proportions, right? That's the idea of relative abundance, right? This population has a different number of individuals that comprise it or represent it versus this population, or this population has more individuals relative to the total number of individuals in all of the different species of that community. So relative abundance is the percent composition of an organism of a particular kind relative to the total number of organisms in that area. Again, that plays into this understanding of evenness. In communities, if species have more similar abundances with one another, you have greater evenness in that community. So evenness is a measurement of equitability among the species numbers or individual numbers among species in the community. And to calculate relative abundance and more specifically evenness, we use an index called the Pelouse Evenness Index. This indice will compare the evenness component of species diversity in a community, which is this H prime value, which serves as the calculated Shannon index value, which we'll talk about in a moment. But is comparing this evenness component of species diversity against the maximum amount of evenness possible given the same species richness. Right? So this value here is representative of basically the maximum amount of potential species diversity under maximal equitable conditions, right? So if you had the maximum number of species, or based on the number of species that were occurring, you could have the highest level of species diversity because each one was occurring at the highest evenness value, meaning each one had the highest similarity of abundance possible, or that could be achieved given that number of species, or given that species richness. So when you calculate this value of evenness, the values will range from 0 to 1. So your J value that you calculate, J ranges from 0 to 1 or 1.0. And 1.0 indicates maximum evenness, right? So if you calculate your J-score or J value, you get a value of, say, 0.968. You have a pretty high level of species evenness in that community, right? So what that's meaning, right, that maximum evenness, it's basically that value is saying that that habitat has similar abundance values for each species present, right? And because of that, that habitat is said to have evenness or greater level of evenness. Now, kind of moving away from the concept of species evenness and returning more to the notion of abundance. When we talk about abundance, the abundance of species, it's referring again to how many individuals represent or constitute a particular species. So you can use it to refer to how common or rare a species is relative to the other species in a defined community. Now, in general, most species are moderately abundant in a community. Few are very abundant or extremely rare. But abundances and abundance patterns in general can be skewed, meaning there are some that are a little more abundant than others, or maybe rare and underrepresented compared to the majority of the community. Now, one way to deal with what we call skewed abundances is to transform them using logarithms. And this was actually visualized and came into being by Frank Preston in 1948. He came up with the notion of the log-normal distribution of species abundance. What he did was he constructed abundance intervals, with each interval being twice the preceding one, and then graphed the number of species within each of those intervals. And that created this, what he called the log to abundance scale. And when he plotted abundances using that log 2 scale, he observed that the patterns of abundance in many communities followed this normal distribution of species, hence the log-normal distribution. For many communities, a log-normal distribution provides an accurate description of the abundances of species in a community, and therefore it's frequently used to model the relative abundance of species in communities. And there are other mathematical models that are used, but that's the most frequent that is used. Other models include log series, geometric series, but again, the log normal distribution is the primary frequent one that is utilized for mapping out or plotting out abundance values for species in a community. So the distribution itself, you can just think of it or consider it as a statistical expectation. And it's important because it allows us to predict the distribution of abundant species. In theory, this distribution allows an individual to determine the total number of species in a community, including those that may not actually be sampled, that may be missed. So these are looking at these actual log normal distributions, kind of the skeleton and the bare bone structures of the distributions. Preston, when he created these, he again graphed abundance of species in collections as frequency distributions using these doubled octaves per se that served as bins for plotting abundance. And when he created these he used several data sets. He worked with breeding bird surveys, moth collections, some mammalian studies, and using these data sets and creating or working with that data, he argued that the species abundances, when they were binned logarithmically using that log 2 scale, followed a normal distribution or a Gaussian distribution. And part of that had a result or was a result of the central limit theorem. So what this means simply is that, again, abundance distribution is log normal. And he used that to also kind of argue that before using that logarithmic scale, most abundancy distributions, or those frequency histograms, they had this right skew. And he said that that was a sampling artifact rather than a true function of the community. So when we actually plot out abundances using the log normal distribution, we can represent communities using this histogram setup with abundance categories on the horizontal axis. Again, these are those binned logarithmic scales as created by Preston or denoted by Preston. And the number of species in each category situated along the vertical axis. Now, looking at this, all right, how do you read this? Well, it's very, very simple. Very common species, like say we had house mouse or meadow vole, they are situated in the right-hand tail of the histogram. Your rare species, like most of our large carnivores, large predators, are more in the left-hand tail. In a well-sampled community, most of the species will occur in the middle of the distribution along this peak. And that's because most species are moderately abundant in communities. And again, that's indicated or substantiated by the peak of the normal distribution curve, or this Gaussian curve, the bell-shaped curve. Now, sampling has a role to play. Incomplete sampling, or restricting sample size so that it remains very small, often causes species to be missed. Even with extensive sampling at times, some species will be missed because there may be very, very rare species that remain undetected, or as Preston coined it, remain veiled. Now, when you start off samples, those early samples reveal common species, right? The majority of your species, which are relatively abundant, moderately abundant, whereas those rare species will remain hidden, and they remain hidden behind what Preston coined as the veil line. This veil line is the point at which rare species cease to be sampled. And he noted that more intensive sampling or increased sampling effort would aid in unveiling rarer species. So looking at this figure here, right, the number of species plotted for different abundance intervals are being shown, with each interval, as far as your abundance interval, being twice the preceding one, so creating that log2 scale. Now the proportion of the graph in this red color that's left to, or to the left of this veil line is theoretical, right? It's depicting those species that are expected to be present but have very low abundances. And likely their low abundance levels prevent them from actually being represented in a sample that a researcher takes. Now, additional sampling, right? If you take a larger sample or continue to increase your sampling efforts, that causes this veil line to shift to the left, right? So less species become cloaked by that veil line. As you increase your sampling effort, it's likely that you are able to sample more rare species, which increases their abundance values, or the abundance values that you record for each of those species, as well as increases the abundances recorded for each of the species already observed, right? So in general, the entire distribution is shifted, right? Your veil line is shifted to the left, which causes the entire distribution of the observed abundance values to be shifted to the right. You get more of this right-tailed skew. Now this plays another important role, right? It doesn't necessarily just highlight the fact that you have to use multiple samples or intensive sampling techniques or large sample sizes, but it again can be used to kind of as this predictive measure, right? Where deviations from a log normal distribution could be used to estimate the completeness of a species inventory. And you can also use those deviations to estimate how many more species might be found if you increase sampling effort or were more intensive with sampling. Now, kind of going along, again, this thought process of, or this notion of sample size and how that affects abundance. When you look at most of your log normal distributions, you rarely will get something that looks just like this. A lot of times you are getting something more along the lines of this, where you don't see the entire bell curve, or one of these plots, again, where only a portion of that bell-shaped curve or Gaussian distribution is present. Looking at these two in particular from one another, you can see that this plot here has more of that bell curved present as opposed to this one. But in general, again, both of them, a portion of that curve is missing if you compare them to these plots that are shown here. Much of that difference between these curves results from a difference in sample size, right? And again, this is something that was suggested by Preston and has since been proved. Essentially, given that species toward the left side of the x-axis are increasingly rare, right? So again, most of your rare species will sit towards the left-hand side of the horizontal axis. It's likely that those species would be missed in a random species sample. But if you increase sample size, the likelihood of collecting rare species in a way that accurately represents their abundance increases. And as you do so, you also increase the abundance values of the other species that have been collected. And this creates or allows a more normal distribution to become visible. More of that bell-shaped curve is able to become present, right? So looking at these two plots here, these are looking at the distribution of different moths in two areas. This is looking at a sample of moths from Lethbridge, Alberta. This one is looking at a sample of moths from Saskatoon. Now, the sample of moths here from Lethbridge, Alberta, appears to come closer to a full curve. Now, why might that be? Well, what have I been ranting about for the past, probably 15 minutes? Sample size, right? While the sample of moths in Saskatoon, it contained approximately 87,000 individuals. And those 87,000 individuals belong to 277 species. On the other hand, you look at this sample from Lethbridge. This contained an incredible 303,251 individuals belonging to 291 different species, right? So what this shows is that smaller samples result in only a part of that bell-shaped curve being revealed. And only a small part of that bell-shaped curve being created that's characteristic of a true log-normal distribution. And ecologists have found that, again, the more you sample a community, the more species you find. If you only use small samples, you will only count or find common species. A great deal of sampling effort is required to begin detecting rarer species and create truer logarithmic representations of abundances through communities. So that's really the in-depth look at evenness, and then delving even more into an aspect of evenness being relative abundance, and considering what abundance is and how you can go about calculating abundance and visually depict it graphically, but also how you, as a researcher, as you may be, as a scientist, how you can consider the effects of your actions as far as sampling, how that may affect abundance values if you're calculating that. So moving away from, let me get this out of here. Oh, stop it, what did we do? I feel like I should be far more technologically capable than I really am. See, how do I get this over? Oh, that's doing that. Okay, now it's better. All right, so I don't know what just happened, but we're back on track. So moving away from how we calculate abundance and evenness, into looking how diversity may be calculated. There are many indices of species diversity that have been developed. In fact, over 60 different indices exist and are used to calculate diversity. There's two types of indices. There's dominance indices and information indices. One indice that is commonly used in research is the Shannon-Weiner index, or the Shannon index. This is a type of information index. So information indices are based on the rationale that diversity in a natural system can be measured in a way that is similar to the way that information contained in either code or messages is measured. Almost like that, basically measured amounts of order in a system in bits of information. Kind of by analogy, if we know how to calculate the uncertainty of the next letter in a coded message, then we can use that same technique to calculate the uncertainty of the next species to be found in a community. Right? That's the whole logic behind these information indices, and that's how this Shannon index operates. The Shannon index is a great index, and the kind of the reason why it's used so frequently is because it accommodates rare species, or the aspect of rarity in species. Even rare species with like one individual will contribute some value to the Shannon index. So if an area has many rare species, their contributions would be accommodated for. Now, the values of the Shannon index for most of your real communities in nature are often found to fall between 1.5 and 3.5. And the value that's obtained from a sample by itself is in and of itself of no significance. This index will only become useful while comparing two or more sites. Right? So it's more of this comparative measure between sites or between habitats. Now you calculate this index value, or this diversity measure, which is represented as H prime, by taking the sum of the proportion of the total sample belonging to the nth species, or ith species, right? Some number of species, whatever number of species you have total in your sample, right? So if you had 5 species, your PI value is going to be the proportion of the total sample belonging to that ith species. So you take the sum of the product of that proportion times the natural logarithm of the proportion of the total to find your H prime value, or the Shannon index diversity measure. You can also use this formula here, the sum of, again, your proportion of the total times the natural log of pi, or the proportion of the total. It's basically the same thing, same formula, just written 2 different ways based on how you write your natural logarithm. If you notice, for both of these, that summed value is negative. So you take the negative or the reciprocal of that summed value to get your true H prime. So if you got a value, say the sum here was negative 1.02, your actual H prime value is going to be basically minus that negative 1.02, so your true value is positive 1.02. In order to calculate the proportion of the total, this is a function of basically taking the number of individuals of a species and dividing it by the total number of individuals in this sample, right? So if you had two, we'll say you had 3 individuals of one species, and in total there's 12 individuals in the sample. You use that to calculate PI, so PI there would be 4. Okay, come on, computer. There we go. So this is an example showing, again, how you can calculate your Shannon index. Here we've, you find the proportion relative, so the number of individuals of a species of interest divided by the total number of species will give you your proportion or PI value. Then you can take the log, the natural logarithm of that proportion, and then you multiply that value times the original proportion that you got to get this PI times natural logarithm of PI value. You sum all of these values together in this column, that gives you that total value, which is then going to be the inverse. So the basically inverse of or reciprocal of this negative 0.237 will be 0.237. So if you end up with a sum that's negative, it's going to be truly positive because this formula is the negative sum of that PI log PI value. Now, with these values, you get values that generally range between 0 and 1.0. Now, the larger the H value, your H prime value, meaning the closer it is to 1.0, the greater that diversity level. So again, if you have a value that's 0.0967, you're going to have an indication that that community that you're calculating or evaluating is of high diversity, has a high level of species diversity. So this is comparing, again, if you're looking at just one community, that index value has little to no substance to it or importance. It's really of importance when you're comparing between communities. Right, so here we have two communities, community A and community B. Community A has 2 species. Species 1 has 99 individuals. Species 2 has 1. So let's calculate species diversity. We're going to calculate species diversity using Shannon index. Right, so using the formulae that I've provided before. Now what you do is you basically will calculate that H prime value for each species individually. Right? And you can do that by taking the basically the absolute value, or the negative of your pi times the natural logarithm of pi for species 1 plus the pi times natural logarithm of pi for species 2. So here we would have 0.99 times the natural logarithm of 0.99 plus 0.01 times the natural logarithm of 0.01. So here's for species 1, this is for species 2. When you calculate that out, you get an H prime value of positive 0.056. So this is, right here, this is this diversity measure, or species diversity measure for community A. Now we can also look at evenness, right? So evenness is going to tell us, it's not going to give us a measure of specifically diversity. It's going to tell us how, based on the number of species we have, how evenly distributed are the individuals in total, right? So of the total number of individuals we have, how evenly distributed are they amongst the different species? So do each species have a relatively similar abundance to one another as far as the number of individuals? And we'll calculate that, again, using that Pelou's evenness index. So here, again, that formula is H prime divided by H prime max, right? So the max, the maximal value, that's what I want to say, the maximal value of evenness that we could possibly have in this community is a value of 0.693. We take the current diversity value that we have divided by the maximal diversity that we could have given this community, and that gives us an evenness value of 0.081. So in general, we have pretty good diversity, pretty good evenness. Now you compare that to community B. Species 1 has 50 individuals, species 2 has 50 individuals. So when you calculate the H prime value, you get a species diversity level of positive 0.693, which is far, far greater than community A. We now see that community A has a pretty stinky species diversity. And the species evenness for this community is 1.0 because it's a 50-50 split. Same number of individuals in species 1 as there are in species 2. So because this community has a far greater species diversity value in general, the diversity of this community is far greater in community B than it is in community A. But we can also just look at the evenness, and from the evenness alone say, if we didn't calculate diversity, we could say, based on evenness, this community is going to be more diverse or have greater species diversity because both communities have the same number of species, but the individuals representing those species in community B are far more evenly distributed across those species than they are in community A. Community A, almost all species except for one, well, all species except for one, are a species 1. So another major index type that's used, besides, again, Shannon is the major one, but another one that's used is the Simpson's Diversity Index, actually properly stated, Simpson's Diversity Indices. In general, this index is a measure of diversity which takes into account, as with the Shannon's index, both richness and evenness. and it's a type of dominance index. Dominance indices are weighted toward the abundance of the commonest or commonmost species. And it does not quite well take into account rare species of a community like your information indices. And that's a disadvantage of dominance indices in general. So a disadvantage of the Simpsons index, being that it's a dominance indice, is that it's heavily weighted towards the most abundant species, and it does not accommodate rarity. So the addition of a rare species with one individual, for example, would fail to change the value of this index. It would bear no weight. So when you're using diversity indices in conservation biology, this has limited value, right? Because especially if an area, I should say, has many rare species with just one or three individuals. Now, the term Simpson's Diversity Index, as I said before, was more properly stated as Simpson's Diversity Indices because it can actually refer to any one of three closely related indices. You have the Simpson's Index, Simpson's Index of Diversity, and Simpson's Reciprocal Index. Each three represent the same biodiversity, but they are interpreted differently and actually do provide a different measure as far as interpretation. So it's important to ascertain which index you are using when you are making these comparative studies of diversity. And we'll talk about each one. So get out of here. So the first one is the Simpson's index, which is denoted as just D. This is used to measure the relative biodiversity of a given community or between communities. More specifically, it measures the probability that two individuals randomly selected from an infinitely large community or sample will belong to the same species or same type, same family, same genus. And there's two versions of the formula for calculating this index value. Either one is acceptable, but you have to remain consistent. Right? You can either calculate it using the sum of the total number of organisms of a particular species divided by the total number of organisms of all species squared, or you can take the sum of the total number of organisms of a particular species times the inverse, so little n minus 1, divided by the total number of organisms of all species times the total number of organisms minus 1. Either one works, you just have to remain consistent. So try not switching between one or the other in this common study. Now, the value of D ranges from 0 to 1. With this index, 0 represents infinite diversity, and 1 represents no diversity. So the larger the value of D, the larger the value of D, the lower the diversity. Now that is neither intuitive nor logical. So to get over that problem, your D value is often subtracted from 1 to give what is called the Simpson's Index of Diversity, denoted as 1 minus D. In this case, the index represents the probability that two individuals randomly selected from a sample will belong to different species. So the Simpson's index that I just discussed before, again, is the probability that they will belong to the same species. This diversity index, the Simpson's index of diversity, 1 minus d, is the probability that they will belong to different species. So the value of this index also ranges from 0 to 1, but now the greater the value, the greater the sample diversity, which makes a lot more sense. So you often see this used more frequently than just the Simpson's index, so just D. Another way of overcoming the problem of the counterintuitive nature of the Simpson's index is to take the reciprocal of the index, right? So that's this value called the Simpson's reciprocal index. And this can be used to measure the relative biodiversity of a given community and also compare communities to identify different intrinsic qualities. Now, the value of this index starts with 1 being the lowest possible figure. So in this case, 1 would be representative of 1 species or 1 individual. This figure would, again, represent just that 1 species, and that would be that lowest possible figure for the scale. The higher the index value, the higher or greater the degree of biodiversity within the community. Right? So a higher index value, Figaro, stop. That is a piece of wood. Yes. Go find your squeaky toy. No, not the Pop-Tart wrapper. Okay. So, what was I saying? So the higher the index value, again, the greater the degree of biodiversity in that community. Now, the maximal value that you can have is equivalent to the number of species that occur in that sample. So, for example, if there are 7 species in the sample, then that maximum value is 7. But no matter how many species you have, the lowest possible figure will always be 1. Now, as I said before, the Simpson's reciprocal index can be used to compare communities to identify intrinsic qualities. A high value suggests a stable site with many different niches and low competition. So you have high richness, high evenness. A low index value represents a site with few potential niches, where only a few species may dominate. So there's low richness, low evenness. And your index value can change. So it's not going to remain constant. It may change in response to an ecological disturbance, which could range from any sort of anthropogenic activity to natural disasters. All right, so let's do an example. In this example, we'll work out the value of D, so our Simpson's index value, for a single quadrat sample of ground vegetation in a woodland environment. Now, obviously, this would not be a great study at all. This would be an epic fail because sampling with only 1 quadrat would not give you a reliable estimate of the diversity of ground flora in the wood. You would need several samples to be taken and then pool that data to get a much better estimate of overall diversity. But for the sake of this example, we're just going to use a single quadrat. So calculating or using this information these are the number of individuals per species of ground vegetation. We can then take that number and times it by the number of individuals minus 1. If you sum all of these values, that will give you your n value, your small n, right? So the sum of our little n times little n minus 1, or the number of individuals times number of individuals minus 1, is 64, right? We're going to then divide that by the total number of individuals times the total number of individuals minus 1. So our total number of individuals in this entire quadrat sample is 15, right? So we then take 64, that's the value, the sum that we've gotten for our individuals, for individual species, divided by 15 times 14. So 15 times 15 minus 1, and that's going to give us a Simpson's index of 0.3. So based on that, we can then say, all right, Simpson's index of diversity is going to be 1 minus that d value. So 1 minus d, or 0.3, is going to give us a Simpson's index of diversity of 0.7. And then our Simpson's reciprocal index, which would be 1 divided by d, so 1 divided by 0.3, will be a value of 3.3. So with this, I like looking at the Simpson's index of diversity. So looking at this value of 0.7, again, you're going to compare that to the value range between 0 to 1, where 1 is representative of greater diversity. So by having a value of 0.7, you can say, okay, this area has a relatively high species diversity. And then you can use that to compare with other communities. Maybe you take a single quadrat of ground vegetation from a neighboring community of in one of your fir forests, and you get Simpson's index of diversity value of 0.2. You can then say, all right, the diversity of that first community, this community here, is greater than the diversity of that second community that was sampled. All right, so as I was saying before, when we're considering areas that are, or determining what community is more diverse than the other, you have to consider both species richness and species evenness. Right? Communities with even distributions of species are more diverse than communities with greater richness, but skewed distributions. Right? So in this example here, both communities, community A and community B, contain the same 5 tree species. But because community B has greater species evenness, right, there's more equal number of individuals per each species found in the community, it has a higher diversity than community A. Community A, again, they both have the same species, but the evenness of individuals in the species of this community is far lower. Most of the individuals are of this weird little brown fur species rather than the other individuals, the other four. This one has a more equal representation of individuals within each species, therefore is more even. Now, there are, as I've mentioned throughout, kind of non-directly throughout this lecture video, there are a variety of factors that influence diversity. We talked a little bit about this area size, right? So the larger the area, in general, the more species based on the species area relationships and the species area curve. Latitude actually seems to affect diversity. In general, there's a pattern of a high species present or greater number of species present along the equator, with diversity dwindling off as you move towards the poles. And this pattern is actually known as the latitudinal diversity gradient, or LDG for short. Climate is another factor. In general, warmer, moister regions, primarily those along the equator, have greater biodiversity, as you can see from this image here. Figaro, stop. Oh, I swear he's like a child. But other factors are niche diversity, genetic diversity, introduction of new species. It could be an invasive species that's introduced, which may drive out or cause the extinction of other species, and that's going to affect diversity, as well as anthropogenic activity, which will cast various disturbance events, which may or may not cause extinction of species, but also could allow for the arisal and colonization of new species. So honing in on some of the more interesting factors, or some interesting trends that you see with diversity. Many studies show and have shown this positive relationship between environmental complexity and species diversity. In general, species diversity increases with environmental complexity, or heterogeneity. Now, why is that? Well, the reason for such is because a higher environmental complexity introduces a more diversified environment that contains more niches. Right? More niches will increase species diversity because there are more places that are available for species to occupy. So greater availability of habitat space, also greater opportunities for colonization by different species, because you have more niches, will allow for more species to occupy and colonize, and therefore results in higher species diversity. Now, this was a concept that was first quantified by an ecologist named Robert MacArthur. He was the first one to actually quantify the relationship between species diversity and environmental heterogeneity or complexity, when he studied the diversity of warblers throughout a variety of forests in northeastern United States, Florida, and Panama. And when he was studying these warblers, he found that the different warbler species all fed in different parts of the forest canopy. And this goes back to that concept of resource partitioning that is discussed in the early lecture video regarding the objectives and major concepts of Darwin's theory of natural selection. It's all this act to avoid competitive exclusion. Now, MacArthur quantified the diversity of species and the complexity of the environment using the Shannon's Index. And then he measured environmental complexity as a function of foliage height diversity, which increased with the number of vegetative layers and with an even distribution of vegetative biomass among different vertical layers. And he looked at three specific vertical layers. His layers that he looked at were intervals of 0 to 0.6 meters, 0.6 meters to 7.6 meters, and then greater than 7.6 meters. Now, the foliage height diversity that he calculated, like species diversity, increased with richness, right? So it increased with the increasing number of vegetative layers, and it also increased with evenness. So how, in this case with the foliage, how evenly dispersed or distributed vegetative biomass was amongst those layers. Now, he and his colleague and kind of sidekick, John MacArthur, they measured foliage height and avian diversity in 13 plant communities in northeastern North America, Florida, and Panama. And the vegetative communities that they evaluated ranged from grassland habitats to mature deciduous forests. Now, MacArthur found that warbler diversity in general increased as quote unquote vegetation stature increased. So, in other words, plant communities with greater foliage height diversity supported more diverse avian communities or more diverse bird communities. And so he took this information and then went on further to study this relationship in a variety of other forest or habitat types around the globe, other regions of North America, all the way to various regions of Australia. And he found continually this positive correlation between foliage height diversity and bird species diversity. Right. So from those findings, again, bloomed this concept that as you increase environmental complexity, there are more niches that are available for species to occupy, and as a result, species diversity will increase. Another interesting relationship as far as biodiversity or species diversity being influenced by some external factor is this notion of disturbance. A man named Joseph Connell proposed that high rates of diversity were a consequence of a continually changing environment or continually changing conditions. And from that proposition, he created what was called the intermediate disturbance hypothesis. This hypothesis proposes that intermediate levels of disturbance promote higher levels of diversity, right? So in this notion, the highest diversity of a species in an ecosystem is maintained by some level of disturbance. And that level of disturbance is intermediate, meaning it's falls between frequent and rare in occurrence. If disturbance is too frequent, then what can actually happen is succession would or may fall or fail to develop beyond some of the early phases of succession. So it may fail to develop beyond the pioneer phase, or only species that were tolerant of stress would be able to persist. Right? So you would actually hinder the cultivation of species diversity because of high stress conditions. On the same hand, or on the kind of the other spectrum, I should say, if disturbance is rare, doesn't happen enough, the end stage of succession, which is known as the climax stage, will be established, and diversity would actually reduce because of the competitive exclusion principle. Because at that point, you would have the domination of certain species, and only those dominant species would persist. So at intermediate levels of disturbance, when you have some disturbance occurring, but not too frequently, not too rare, high levels of diversity are promoted. And the reason for that is because there is sufficient time between disturbances for a wide variety of species to colonize an area, but not enough time to allow for the competitive exclusion principle to set in.